TTC, GAA, TTC. Agreed? And you see that we made a cut between the G and the A, the G and the A, correct? So what I did on the next slide, here I made the cuts for you. And then I pulled the strand apart. Here's a strand that's going to have an overhang, and here's the other little strand that's going to have an overhang. So I pulled them apart on the next slide. Let's take a look at that next slide. Did you see me pull them apart? Right? So this is what they look like when they're together. That's what they look like when they're pulled apart. So here's one, and here's the other. What did I do to the ends? After I pulled them apart, I went ahead and labeled the ends. And these ends are labeled correctly because there's a 3 here, there should be a 5 here. If there's a 5 here, there should be a 3 here. And I even labeled these ends. You see that? And by the way, if this is circular DNA, do you agree that after the cut is made, now this piece of DNA looks like that? Agreed? Now here's a question for you. Whether you're talking about this one or this one, can you tell me which end is sticking out the farthest?
The 5'9".
Therefore, this is an example of a 5' overhang. Does that make sense? Okay.